Good evening, everyone, and welcome. We are super excited to invite you to or have you join us for our February Science Cafe. It feels like we were just here a minute ago for our January Cafe as we were kicking off 2025. So already here we are, it's February, and we are very excited to be talking with you this evening about the health of our vascular system and its role in healthy aging. So that's a topic that I'm sure we are all interested in. I know I am, and we are very excited to have two experts on the subject with us this evening, and I will introduce them in just a moment. I want to just say a quick couple of comments about some housekeeping details. So those of you who were with us last month, you know that we had some guest visitors that we maybe wish that we didn't. So we're trying to kind of clarify our process here a little bit. We ask that it looks like all of you have done a great job of including your name on your sign in, which is very, very helpful, and I think helps to contribute to our sense of community here. We've also added closed captioning. So if that's helpful for you in terms of being able to follow along and hear or comprehend what's happening with our speakers, you can turn that on in the lower bar on your screen. And I find it actually, it's a wonderful tool. So feel free to explore that and some of the other new tools that are in Zoom these days, and we hope that that will help contribute to a really active discussion. So with that, I want to also make give a quick shout out to Victor and Patricia Hand, who are hosting a watch party for us this evening. So welcome to all of you, and we look forward to having some great conversations with that group this evening as well. But now I want to just take a moment to welcome Dr. Anastasia Paulmann, who is joining us this evening as we talk about the role of the vascular system in kidney regeneration, kidney health, and also just, health in general, right? The role that it plays in healthy aging. So a little bit about Anastasia. She is a physician scientist who is from Hanover Medical School in Hanover, Germany, and she is just wrapping up a 3-year research program here at the MDI Biological Laboratory, and she is doing that work in the lab of Dr. Hermann Haller, who is also with us this evening. So I think many of you are familiar with both Anastasia and Hermann and their role in this exciting area of research. So I'm going to ask Dr. Haller to kind of kick us off a little bit as we embark on this conversation. And Dr. Haller, I'm wondering if you can set the stage for this conversation by telling us a little bit about the vascular system. What is it? What does it do? And what is the role that it plays in maintaining health in the body?
Yeah, I'm happy to do so. It's a great pleasure to do this Science Cafe together with Anastasia. Thank you very much, Terry, for the introduction. Could I have the next slide, please?
Slide is coming. I hope everybody can see the presentation.
Yes.
So aging and vascular health, as Terry already mentioned, new insights from African turquoise killifish. And please do not hesitate to ask questions either in between or keep your questions at the end, because both Anastasia and I understand that this will be hopefully not too complicated, but complicated. And we are looking forward to your question. Anastasia, if you could click me through the next slide. So when we talk about the vascular system and aging, what we are actually talking about is the microcirculation. And since not all of you are familiar with the microcirculation, to be honest, a lot of doctors are not really that familiar with the concept. On the left-hand side, you have the circulatory system. We have the heart, and then from the heart, the blood is pumped through the large, very important arteries. And all these arteries then go into the periphery to different organs. And in order to provide the organs with the nutrition, with oxygens, to remove waste, the arteries break into smaller and smaller capillaries. It's like in a tree. And these capillaries are very small. They only have one cell layer, and the whole exchange of oxygen supply, nutrition, energy, removal of waste is taking place, and this is shown in the middle, the artery going into these tiny capillaries, and then the basis of how our organs live, how the skin, how our muscle is supplied. This is all the microcirculation. And you see how delicate this is. On the right-hand side, the microcirculation of the brain, the microcirculation of the heart, and of the kidney. Unfortunately, it's very difficult to assess. So when we measure blood flow normally, we measure blood flow in the large arteries, and the microcirculation is definitely under-researched. And when you look at the importance, we need to know more about this. And this is what we're doing. Give me next slide, please. On the left-hand side, you see what the microcirculation for us looks like, all these cells which are involved here in the capillaries, and what you see on the bottom, blue, the vascular endothelial cells, and this is what we are interested in. And we have 3 areas of research at MDIBL in my lab. How does the microcirculation form? How do we grow new blood vessels? And this is what Corey Johnson is doing using stem cells. How does the microcirculation stay healthy? This is what Yannick Becker is doing. And we have told you about the glycocalyx, which covers these endothelial cells. But next slide, please. The third area, and this is the topic of tonight, is how do we prevent the microcirculation to deteriorate during aging? And on the 2 photomicrographs, you can immediately see how important this is, because on the left-hand side, you see a healthy microcirculation, this intricate system, as I said, like a fresh tree. And on the right-hand side, you see that we lose these blood vessels during aging. And this is what Anastasia will talk about. You can ask her all the questions, not only about the microcirculation, where she's now the expert, but she has transplanted kidneys, she has treated patients on dialysis, is taking care of patients with acute and chronic renal failure. So do not hesitate to ask us questions about the conditions after the talk. But now, Anastasia, it's all yours, and we're looking forward to the exciting story of aging, of microcirculation, and what you have found out.
Thank you very much, Professor Haller. I hope everybody can hear me well. Thanks again, everybody, for having me on tonight. Thanks, Jerry, again for the invite. And I would start off. You probably heard that I'm interested in researching aging. I feel like the rings of a tree are a great representation of aging and a true biological clock. Every ring reflects the environmental conditions of that particular year. May it have been a drought, a fire, or a year of abundance. It reflects into every year. And I feel it's very similar to how we or how our lifestyle and our diets affect us as we age. But what's the technical definition of aging? So if you were to look into a dictionary, you would see that aging is the inevitable, time-dependent decline of physiological function in various organ systems due to accumulation of cell damage. So I feel like this sounds a little bit depressing, that it's inevitable and time-dependent, but what if I can tell you tonight that we are hoping it may not be as inevitable as you might be thinking? So you have already seen this picture. Basically, what we know and what we have researched is that aging leads to a loss of tiny blood vessels. And you can see on the right side the microvasculature and the large vasculature of a mouse kidney. And on the left side and on the right side, you see how that looks like in the old animals. And this process we call microvascular rarefaction. So again, loss of tiny blood vessels. So our hope in my research aims that if we can preserve the vasculature even as we age, we would have an anti-aging treatment. So keeping the blood vessels young helps slow aging. That's the overarching theme of my research, and I'm using a very exciting model to study this. So we are using the African turkey's killifish. Some of you might remember my talk from last year that the African turkey's killifish is the shortest lived vertebrate that we can keep and house in a lab. Again, on the left side, you see a young killifish with its vibrant colors. And on the right side, you see our old killifish that are a little bit grayish. You may already see how they're a little bit more hunched over than the young animals. And the nice thing is when we do killifish research is we typically see all the classic signs of aging that we also find in humans. So here you see on the top young killifish and in the middle and bottom older killifish. And we know that the killifish undergo these typical aging signs. So they lose muscle, they lose bone density. They even have not only loss of color, but also loss of memory. So killifish are, for example, also a model of dementia, which don't ask me how you measure a demented killifish, but we're looking at something a little bit different tonight. So when we set out to do our work, our hypothesis was to link lifespan to blood vessels. And we assume that if we have young animals with a healthy microvasculature and they undergo the regular aging process and turn into old animals with signs of lost health, they should lose microvasculature. So when I set out to do this work, the first question we asked ourselves, what are exactly young and old killifish? What are the time points for that? So the way we determine this is we assess it based on lifespan. And this is something that can be done in a killifish, but it's a little bit difficult to translate it into a human. So for example, a killifish is young when it's about 6 to 8 weeks old. A killifish is old when they're over 18 weeks old. The reason we chose those time points is because the young killifish have basically just matured, so they are able to reproduce. You can think of this as somebody in their 20s maybe. An old killifish, on the other hand, is somebody that we said, well, if 50% of the entire population has died at that time point, this is when we would say it's an old killifish. Maybe to make it a little bit easier and to be able to visualize this, what I'm going to be showing to you today when I'm be comparing young to old killifish is a little bit as I'm comparing myself to Professor Haller. I have to admit, Professor Haller, you're a lot younger, especially biologically, than our young old killifish. And I have to admit, I'm a little bit older than young killifish, but you get the idea. So we're comparing young to old time points. Now, when we know who is young, who is old, now is our hypothesis the basis of our idea that old killifish lose microvasculature? Is that even true? So this took a long time to figure out, and I'm happy to share with you that we can see this process. So here you see an image of a young killifish heart. You see, I hope you can see my mouse. Yes, so this part of the heart is what we call the ventricle. This part is what we call the atrium, and this is the so-called bulbus arteriosus. So the killifish, other than the human, does not have 4 chambers, but 2 chambers. And you can see in this image, this bright green color, that's the blood vessels. And in blue, you can basically see the background, the entire organ. And the way we took this image, it is basically an entire killifish heart. And you can see the scale bar over here. The entire heart is just 1 millimeter in size. It's very tiny tissues. So with tiny tissues like that, we can basically just place them on a slide and image them in 3D. And this image is just a representation of this entire architecture. And now that you have seen the young heart, this is what an old heart looks like. And you may already appreciate with the first glance, well, there's a lot less blood vessels in the old heart. And the heart overall is much bigger. So this is very similar to what also happens in human. And if we do science, of course, we also want to have numbers to back up what we see visually. So for example, if we look at the regions where we can see the vasculature, you may be able to appreciate on these zoomed out images how much less vasculature is in the old animal compared to the young animal. And when we analyze data like this, we have tools that allow us to calculate this, like here represented as white area, compared to the overall organ area. And
Anastasia, can I just interrupt for a second? We have a question. I'm going to call on our folks at the hand watch party. They are excited to ask a question. So
yes,
go ahead. Can you hear us, Sarah?
Yeah. Yeah, so the question is, it goes back to before when you were showing the graph of the aging of the killifish. Do killifish lose the ability to reproduce at a certain age? Yeah. You know, do they, as they age, do they lose the ability to reproduce? Oh, so
I'm so sorry. Could you repeat the last part of your question? I understood that it's about the old killifish.
I think she was asking, do killifish lose their ability to reproduce at a certain age?
Yes, it definitely goes down. So the loss of reproduction is also a typical hallmark of aging. I can't tell you for sure whether fish undergo menopause. I don't think so, but we see that the females are not able to lay as many eggs as when they're young, and the males are also not able to fertilize these eggs as efficiently as they were young. Actually, when we do interventions and we try to make our old animals healthier, the rate of fertilization or the reproductive health is one of the parameters we measure to determine whether we have increased the reproductive health of our animals. I hope that answered your question.
Great.
Okay, so now that I have shown you the blood vessels in the killifish heart, you might have heard that me and Professor Haller are actually kidney doctors. So of course, we are not only interested in seeing this process in the heart, but we can also see it in the kidney. And here I wanted just to point out, like, you not only see that the vessels become less, but the vessels that are there are smaller in diameter and they are more fragile. And again, we see the same thing in the kidney. So here you see an image of a young kidney and now compare it to an old kidney. So again, you see a lot less blood vessels. And we not only see this in the heart and in the kidney, but also we see this in the liver too. And this is just an example of like a figure on the image that we would, for example, present on a scientific poster or a scientific presentation where we have quantified in numerous animals, these are these tiny dots, and we have made averages where we can see over 3 organ systems that the young animals have more percentage vessel area than the old animals. So at this point, I'll make a tiny little break and see if there's any questions.
And just a note, if you have a question, you can use the raise hand feature on Zoom, or you can put your question in the chat, whatever is easiest. These are really dramatic images that you shared with us, Anastasia. It's really exciting. So we look forward to hearing the rest of your story. Thank you.
Yeah. And even just taking those images, like something like this, you can imagine I have this for many, many fish and many animals that we sacrificed. And the ability to be able to see this in the killifish, it was very, very difficult to do. So I was very happy that we had really great support, for example, from Hannah Sommers, who helped made these pictures, and Corey Johnson, who also works on another project. And it's basically like also a strength of the MDI-BL, I think, to be able to make things like that, because this is where the expertise of the entire lab comes together. And this type of work has never been done in the killifish before. So we are very excited that we have something that we know happens in mice and humans. And now we can prove, well, it happens in our aging fish as well. So we were very happy to be able to see that.
That's cool.
Anastasia, there's a question about the brain. So you have shown very nicely kidney, heart, and liver. And the question from Ben is the same observation seen in the brain of the killifish?
Well, thank you really very much, Ben. I have to admit, I have to do that still. The reason I haven't really taken out brain of the killifish yet is because the technique to take out and preserve the brain is quite different from their internal organs. But I would assume that it's the same in their brain. We do certainly plan on showing that in the future. Okay. If there's no question until this point, I think to make like a short summary, what I've shown you right now is what are young animals, what are old animals. Young animals have a healthy looking vasculature. Old animals don't have so much of a healthy looking vasculature. Now, remember, our main idea is that if we want to increase lifespan, and not just the years you live, but the quality of your life. So I'm really interested in increasing health span, the time you spend really being able to do everything you were able to do when you were young. So optimal longevity. And our idea is that if you live a good long lifespan and a good health span, that comes or is associated with a very good microvasculature. And one of our ideas how we might do that is, I'm sorry, we're MDs, is of course a pharmacological intervention. The pharmacological intervention, which I have tried out first to see if we can see this effect, is a drug which is called SGLT2 inhibitor. So what does this word mean? Horrible word. Sodium glucose linked transporter 2. Probably doesn't make it better for you. What does this mean? Basically, it's a transporter in a certain part of the kidney that is that's function is to take up glucose that you, so to say, accidentally pee out. So the reason your body reabsorbs glucose is because many, many years ago, glucose was very important. The only people nowadays that pee out glucose are diabetics. So you can imagine a drug that stops, or basically a transporter that stops you from peeing out glucose, and a drug that inhibits this transporter can be used as an anti-diabetic drug. So the idea behind that is, well, if we have this transporter that is trying to take the glucose back into your blood, and diabetics already have enough glucose in their blood, we stop this transporter and then all the glucose goes out of their blood into the urine. And the level of glucose in a diabetic's blood goes down. This works wonderfully. The interesting thing is these drugs are very old. They're almost historical, I want to say, and they're being synthesized from apple tree bark. So if you've ever heard the saying, an apple a day keeps the doctor away, I'm going to show you today how that relates to our research, actually. So what's so exciting about this anti-diabetic drug? So diabetes has been around for quite some time, and there's also been a lot of drugs implemented to treating it. However, every time an anti-diabetic drug is being released on the market, you have to prove to the FDA that this drug does not accidentally kill off people. It has to have a overarching positive effect. Now, the exciting thing with this particular drug, and I'm gonna start calling them SGLT2 inhibitors from now on, is that when these drugs were used in patients with chronic kidney disease, so patients that are already at a very, very high cardiovascular risk, we were able to see that the drug, in this case, dapagliflozin, which is a typical SGLT2 inhibitor, reduces the incidence of death from any cause. And I just want to drive the point how amazing this was. So you have a cohort of patients that is at very high risk, and you give them a drug that reduces the amount of death in them. So this was a breakthrough which happened, Professor Haller will correct me, I think 8 years ago we saw this the first time. And nowadays, SGLT2 inhibitors are our standard treatment for all patients with chronic kidney disease. If you have chronic kidney disease, or if you have low kidney function or proteinuria, there's absolutely no reason why you shouldn't be having this drug. Now, the interesting thing about this drug is, except for this very particular, it makes you pee out glucose function, we don't really understand how it works. So you can imagine in the community of doctors, when something like this came out, and this is not the only paper showing this, something like a race started to find out, well, what is the mechanism by which this drug does this effect? And the first thing we wanted to see when we did this is, well, if we are going to use SGLT2 inhibitors in a killifish, does that even work? Does the killifish have sodium glucose link transporter here? And what I want to show you here is an image of kidney tubules. So what's a tubule? So you can see these round structures, and the kidney is basically built from many, many tubes, like pipes. And you're basically looking here at a cross section of a pipe like this. So this is, we call it the lumen of this tube. And because it's a very tiny tube, it's called a tubule. So as you can see here in blue, you can see the nuclei of the cells. And in yellow, we can see the SGLT2. And the very interesting thing is that this looks in the killifish just the same as it does in human. So again, another surprising correlation of human and killifish, why killifish is such a good model. Now that we know SGLT2 exists in the killifish, we have also shown and proven that we can use the same drugs we as humans take on our fish. Now, if you remember, our first question, so to say, was, well, how does the lifespan of this fish change if we give it this drug? And the exciting thing we can see is, and I'm going to show you first how to read the next lines. These are two survival plots. So you can see on the left-hand side, survival of female fish, and on this side, the survival of male fish. Up here, you see the probability of survival. So over here is basically 100% of the entire cohort. And down here, you can see the time as the fish basically grow older. And this is what our control animals look like. Now, if we look at the SGLT2 treated animals, we can see quite a difference. And what's very interesting is we can see, especially in the females, how the green curve of the treated females looks so much different from the normal females. We don't quite see this in the males. There could be different reasons why we see this, which we are currently investigating. So this was for us already very exciting to see. And now that we know that our treated animals, especially the girls, live longer, the second question will be, well, do those animals have a better vasculature than the other animals? And hopefully by the next Science Cafe, I can show you those results.
Great. Anastasia, we actually have a question in the chat that's very similar to that. So the question is, number one, do you see that these killifish in the population that you have, that they age at different rates? So are there older killifish that are healthier? And if so, what does their vasculature look like compared to the less healthy older?
It's a little bit, I would say, difficult to tell, because of course I'm also very interested in looking into heterogeneity, like the same as like all humans are not the same, all fish are not the same. So we can't, and I'm just gonna like call it cherry pick, so to say. I don't like choose the like worst looking, the most old looking animals when I do this analysis. But we decide at a certain time point, okay, now all our remaining animals are old, and we sacrifice and analyze all of them to really get a good understanding of the differences between the different fish. Let me go back just a little bit, and I can show you on this overview slide. You can see if we quantify, I hope you can see the dots, if we quantify the amount of vasculature, it's not exactly the same in all of the animals. That's why we always need so many numbers. And the more numbers we have, the tighter our results become. And that's why it's always important to look at all the animals, even the ones that we think, well, these guys look healthy and these guys don't look so healthy, and just compare them to each other. So that's something that's very interesting for us. If I look at the differences between my, let's say, control animals and my SGLT2 inhibited animals, it's a little bit, I'm very biased. Like, I would believe that the SGLT2 inhibitor treated animals move around more and have better color, but I'm afraid somebody else will have to look at that as well.
That's great. So we do have a couple of questions about the SGLT2 inhibitors. The first one is, what if you are not diabetic and are not overweight? Are they still recommended, or there's a particular?
Excellent question. Okay, so SGLT2 inhibitors are right now on the market to treat diabetics, to treat chronic kidney disease, and to treat patients after heart failure. So if you ever had a heart attack, you should be taking an SGLT2 inhibitor. The reason I'm studying them is because I'm interested in knowing if they're anti-aging. So right now, there is not one single medication that is anti-aging on the market. And I think the very first medication that will come out to be anti-aging, if you know the company that's going to release that, like, you should invest in that because it's going to be huge. So right now we don't really know if it's something that really does increase the lifespans in humans. Having said that, you can see the way my experimental design is built right now is we treat our animals over their entire life. So the moment these animals have matured into adults, we've put them on the diet to see the long-term effect of this medication. This will give me a different result, so to say, if we were to start treating them at a much later time point or only for a short period of time. It always depends a little bit on what you want to achieve and what your goal is.
Fascinating.
Can I add something? The question about not diabetic and not overweight. As Anastasia mentioned, chronic kidney disease, this is now the one and only serious treatment strategy for patients with chronic kidney disease. It's still not understood. It may have to do with blood vessels as well. But when you have chronic kidney disease, that means when your creatinine is higher or your GFR is lower or proteinuria, as Anastasia mentioned, this is an indication, has nothing to do with diabetes or overweight.
Interesting. So Anastasia, I want to let you complete your acknowledgement slide here because we have a number of questions, and then maybe we can stop the screen share and we can all just have a conversation with all.
Oh, that would be amazing. Again, thank you so much. Once again, I want to point out that work like this in an animal model that is very new, the killifish is new to the entire research field. A work like that, especially in a field, nobody in the killifish field looks at blood vessels the way we do. And then with a translational focus, a focus of how can we use this fish to help humans, would not be possible if not for an entire team of people and the expertise that is found here at the MDIBL. So something I want to point out is that the transporter I'm studying, SGLT, was actually first cloned here in the lab. Professor Haller will correct me when that was, but it was many, many years ago when it was first discovered here. So that is already very exciting because we have a history for doing this type of research. Again, I want to thank Corey and Hannah for help making these amazing images. I want to thank Matt, Tom, and Lynn, who are part of our tight killifish crew, and of course Sonia, who's working on this project in muscle. And I also want to, for this particular talk, point out James Godwin, who, as you know, is the PI who's working on axolotl, who for this project taught me how to design and make my own antibodies. Because the picture of the SGLT2 I have shown to you is actually an antibody we designed ourselves. So we are still in the process of optimizing and building the tools needed to answer all the questions we have. And with that, again, thank you very much for listening. This is, as you know, our wonderful campus. And this is where I'm staying with my little killifish. And if you have any questions that we may not answer tonight, please feel free to reach out to me.
Thank you so much, Anastasia. So I mentioned we have a number of folks who have questions. We have some that have raised their hands. We have others that are in the chat here. So some of the chat ones have been here for a bit. So I just want to get to those in just a sec. But the first is, how do the levels of SGLT2 gene expression and protein production differ in males and females? You know that?
That's a very good question. So the protein production we are still currently measuring, that I cannot say. The interesting thing I can say is that the function of SGLT2 in itself is not different between males and females. And excitingly, it is not different in young versus old animals. So if there is ever a time point where you're like considering taking the drug, or there is an indication for you to be taking the drug, you're male or you're female, you don't have to worry. The drug will work as effectively whether you're young or old, male or female. At least in the fish.
Boyer, do you have a question?
Yes, thank you. Congratulations, Anastasia. Very beautiful presentation. I'm interested in what is happening to these blood vessels. I mean, you clearly showed us that they're reduced in number, but the blood vessels are composed of various cell types, particularly endothelial cells. And are they becoming obstructed, like with atherosclerosis, or are they becoming programmed cell death? What do you think is the mechanism of attrition of the blood vessels?
Perfect question. Thank you very much. That is actually the next topic of our research. We have a few ideas of how this process is happening, and we have a hypothesis right now. So what we are able to do is we can take these old animals, the non-treated, our controls, and our treated animals, we can take out their kidneys, and we can basically make single cells, and we can run transcriptomics, and we can then compare all the endothelial cells, so the inner lining of all the vessels. We can also compare these two groups looking at the outer lining of all the cells. So these are cells called the pericytes. We can count those cells. So I know that not only do the vessels themselves disappear, but the number of endothelial cells in old animals is less than the number of endothelial cells in young animals. And this process is actually surprisingly more profound in regular females than in males. So this is interesting, because if we treat with the drug, it's the females who benefit more from this process. So we are still a little bit in the process of wrapping our head around where they go. Do they turn fibrotic? Do they die off? And we hope to show this in the future as well.
Again, congratulations, you've got a great project.
Thank you.
There is another question, and I think this goes along with what you just said. It's a question about, is the loss due to the muscular wall or the endothelium? It's only the endothelium. There are no vascular smooth muscle cells involved in the capillaries. So it's only pericytes, and this is another cell type we are interested in, or the endothelium. It's not like in the larger vessels, vascular smooth muscle.
And so there is another question, Anastasia. This one, you may have already sort of touched on this, but the question is whether or not there is a reduction of death between males and females when using the drug. And I think we sort of showed that, but I just wanted you to.
Yeah. So again, a little bit methodics are to know here. So right now, I'm not seeing a difference for the males, but a difference in the females. However, these are still ongoing experiments. We are increasing our number to be very sure that everything we show you then turns out to be true. The other interesting thing is, of course, we are not working alone. So, for example, there are projects and collaborations we have going on with either other killifish institutions and with our colleagues from the Jackson Lab to do the same work in mice. So we have a long-standing collaboration with Ron Costancio from the Jackson Lab, and we do very interesting projects looking at SGLT2 inhibitors in mice. And seeing what I'm always very surprised by is how similar the work I'm getting from the killifish is to the work they see in mice. Mm-hmm. So there are two models that are surprisingly similar.
That's great.
In the clinical studies, it's not clear cut. There have been analysis on that. but in the clinical studies, we see beneficial effects both in males and in females. And there is some discussion when you look into subgroups, but it's not clear cut. So this clear difference is seen in the animal models, but the humans are a more complicated model.
Yeah. And this is something I think we should point out at this point, is that the reason we are so excited for this drug in particular is because we have already proven its efficacy in humans. There's a lot of drugs out there that say they're anti-aging drugs that have shown this in worms and flies and mice, but not in human. So this is where doing really rigorous research comes in. And especially if you look into like these entire anti-aging field, there's a lot of very interesting things out there. And again, it's sometimes very difficult and not always a good idea to translate everything we see in an animal model to yourself. However, in this case, we can do this because it has already been shown and proven.
Fascinating. So we have one other question here about is there an animal model with deletion of the SGT enzyme?
Yes, actually, I saw it this year at the American Society for Nephrology Congress. So there is a knockout for SGLT2 in mice, which is very interesting because they did not look at the same question as I did. So they did not do, as far as I know, a lifespan experiment, but they were able to show very other interesting things involving their metabolism, so how these animals produce energy for every cell of their body, because metabolism overall is one of the things we think is what is driving this process and what all these differences we are seeing comes down to.
It's even better. There are human knockouts. So
True. Yes.
there are patients, and they don't have an SGL2, and you can imagine they're lean because they lose a lot of glucose. They're surviving quite well, and this has not been tested, but they live relatively long. I mean, they're protected from a lot of diseases, so there may be a link, but at the moment, this is only speculation.
Fascinating. So I know we have another question from our watch party group. Sarah, I don't know if you can ask that question or.
Yes, thank you. So thank you for that wonderful presentation. It was fascinating, and congratulations for the great work. I'm not a scientist, so this is pretty basic, but am I to understand that as we age, that this transport in our kidneys is weakening, and that therefore the capillaries and the microcapillaries are not being fed, and they're for some reason that you're still researching are blocked or dying or whatever, and that there are ways to counteract this genetic process of deterioration?
Not exactly. So what we know is as we age, we lose vessels. We know this from mice. We know this now from the killifish. We see it in humans with aging and with chronic kidney disease. Now, one of the reasons we think this might be happening has to do again with metabolism and with glucose. And this drug does something which probably has absolutely nothing to do with the glucose transport I have just shown you in the kidney. But there might be some hidden mechanism how this drug saves the blood vessel. And right now we assume it has to do with how energy is produced, how glucose may not be as I don't want to call it healthy, as advantageous in old age as we might have assumed. And again, this is like a part of research going on right now. So it's very exciting to combine these two fields a little bit, the transporters we see in the kidney and the effect it has on the vasculature. So maybe Professor Haller wants to tell a little bit. I know there's like a lot of debate between nephrologists, endocrinologists, and cardiologists. So people who are specialized in kidney, diabetes, and heart, who actually like finds the mechanism for this drug.
But I think you described it very well. So a lot of focus at the moment in research is going into the question, how do cells produce energy? And as Anastasia just mentioned, when cells are endangered, when they are hit by injury, they change their metabolism, and they do this rapidly. And we're learning in different fields that this is a major source of tissue dying and tissue deteriorating. And so Anastasia has found a mechanism which may turn this around. And there's a question about our work on regenerating and regrowing kidneys. So this is exactly along these lines. We lose in the kidneys which have chronic kidney disease, we lose blood vessels. And with that work, we may be able to stop this process and perhaps even turn this around so that blood vessels reappear. There is one tiny little remark in a study which was published with SGLT2 inhibitors 3 years ago, which may indicate that these endothelial cells actually may have a comeback. And Anastasia, I think, will look into this in the killifish as well. There is a question about the connection with the apple. The apple, I think, Anastasia, was only used by you as an example. It tells you to eat apples, but the inhibitors come from apple bark. So we do not recommend that you actually you start chewing apple bark, but when you chew apple bark, you may pee more because you're peeing out your glucose.
So, Dr. Haller, I'm really curious if you go to your, GP and you say, I want to be on one of these SGLT2 inhibitors, what will they say? Is that a possibility?
Yeah, well, first of all, they are expensive as as new drugs are. They are very important. It takes a while in the medical community. You would envision when a drug like this is out and it has been published in the New England Journal several times that it saves kidney function, heart function, and that it prevents you from dying from these terrible diseases, that everybody would want to have this, but doctors are learning. Both Anastasia and I, we are teaching doctors, we are giving grand rounds on SGLT2 inhibitors as to others. So there's a growing recognition by medical doctors, GPs, that this is important. And all the international societies have agreed on that. So what we are telling you is not just individual knowledge, and so this is a recommendation. Now, the doctor, when you ask them, they need an indication for that, and the indication is not longevity, unfortunately. It is either diabetes or kidney disease or heart failure. Under these conditions, they will prescribe that. So don't go to your doctor and start bothering him or her by, oh, they've told me this is longevity drug. I wanted to, well, no, it's heart failure, chronic kidney disease. However, you can do this also, and you can use the drug in the early stages of that. And you may not know this, but when you go to your GP and check your kidney function, it could well be that you have the early stages of chronic kidney disease, and that's an indication.
That's great. So in the meantime, before we can access these drugs, what can we do to keep our vasculature healthy? What are the things that you recommend that people do to sort of spur this kind of increase in healthy aging? Anastasia, any ideas?
I think what we know now, proven to improve healthy aging overall, is something every patient knows, but it's hard to do. So exercise, I would say number one. Good diet. There's a German saying that goes, you are what you eat. And I think for like your overall health, that is very important. Other things you cannot influence as much. So for example, whether you're a man or a female, that will always have an impact on your longevity. Typically, luckily, women live a little bit longer than men. But the things you can influence yourselves are, don't smoke. That's, I think, very easy to say. If you drink alcohol, drink less. If you don't drink alcohol, that's also good for you. Sunscreen, I think, is one of the most proven things that we can say. And again, the implication on a healthy diet, I think, are very important.
So now you have a second MD on the call. The good thing is that when you go to different MD, different recommendations. So I share everything what Anastasia just said. But a little bit of red wine is actually good for your capillaries because it opens the capillaries, so the blood flow is increased by that. And this is good because training of your blood vessels. A blood vessel is as good as the blood flow. So Anastasia already mentioned exercise is number one. When you walk and you use your blood vessels so that they have to support the tissue, this is what keeps them alive. Without blood flow, when you rest, they think I have nothing more to do. I can go to sleep. There are a couple of other things I strongly believe in, but not all the doctors do. And this is when you use cold and hot showers, you actually stimulate your blood vessels to open up, to contract, to open up again. So these changes also increases the blood flow and makes blood vessels better. So cold is good, or cold and warm together. But overall, it's exercise.
I think if you have a sauna in Maine, you have good chances.
In the winter.
In the winter.
Right, star point, and you jump into the water. That star point is too high because you can't get out and freeze to death. So you should have something which is a little bit better.
That's wonderful. Well, Anastasia, Dr. Haller, Dr. Paulmann, sorry. It's wonderful to have this expertise, and we're so excited about this work because it's really a wonderful example of the importance of finding the right animal model, right, to be able to answer these questions. And so thank you for sharing all this information with us, and we look forward to following you and seeing how the work progresses as you go along. Any other questions before we wrap up? All right, so just a reminder, I think it's there in the chat for you, but our next Science Cafe will be on March 10th, and we will have Dr. Liz Marnek with us, who is talking about pseudoscience and how to spot it. So that is certainly something that is becoming more and more of an issue. How do we sort of know authenticity in science, and how do we do our research to know the information we're getting is accurate? So we hope you will join us for that. And in the meantime, we hope you have a wonderful evening. And thank you all so much for joining us for our Science Cafe. And thank you again to Drs. Haller and Paulmann. We really appreciate your time this evening.